Hello students, today I would like to talk about the central dogma of molecular biology. Now that term central dogma is kind of an interesting one. It's really different than the words that we use to talk about biology very often, but really it just highlights the importance of these processes. So today we should be able to discuss how DNA codes for proteins in the processes of transcription and translation. The first thing I want to do is take a quick look at some data. So looking at all this data here, it's looking at the percentage of our different DNA nucleotides. So we have A, G, C, and T, and then here we're looking at a couple of ratios. So let's just pull something randomly like human, and if we look at the percentage of A and G, C, and T, the thing that we immediately start to notice is A and T are almost equal and G and C are almost equal. And we see that over and over again in life. We see these ratios that are very, very, very close to 1. So because this percentage is similar, that tells us that base pairing is happening. That idea of base pairing, that is the suggested way that DNA is able to hold so much information. It is the basis for how DNA replicates itself. It's also the basis for the processes we'll be talking about today of transcription and translation. There's one sort of exception up here that I just want to point out really quickly. This lovely organism, it is a type of bacteria that's known to carry single-stranded DNA, and so that's why we don't see that exactly close to 1 ratio. The reason I wanted to start by showing you this data is because it's really easy to forget that all of this stuff that we know about DNA is still coming from experimental evidence. And so there's still things being learned about this, and it still follows nature of science. So with that, let's delve in. We know DNA is important. We already know that. We know it replicates itself through DNA replication, but we don't really know how it makes us who we are. And we need to keep in mind that that answer is protein. Protein is what actually makes the structures and chemicals that are the basis of life. The way that happens is DNA is transcribed into RNA, then RNA is translated into protein. This process of DNA to RNA to protein is key. In fact, that process is so key we call it the central dogma of molecular biology. This term dogma, it's defined as established belief or doctrine held by a religion or particular group or organization. It's authoritative, not to be disputed, doubted, or diverged. This definition does not really sound like science. So often with science, we hold on to that idea of error, and this definition doesn't use that. And so the reason we have this definition is really just to highlight the importance of this concept in biology. I'd like to start with a little bit of an analogy. So I like to think of DNA as kind of the recipe book for you. It has all the information for building your cells, has all the information for how your cells are going to differentiate, it has all the information for genetically how to build you. But if you have this whole big cookbook and you actually want to do some cooking, you're not going to bring the whole cookbook into the kitchen because you'll get it messy. So instead, you have just a single recipe copied from the book. That's kind of like the RNA. And then eventually you have what you actually want from this whole process, like a cake. And that's the idea of protein. And so we have DNA going to RNA going to protein so that we can get to this final idea. The first process of DNA going to RNA is called transcription. And the second process of RNA to protein is called translation. Let's break those down. Before we break them down though, let's just highlight again why we care about protein. Remember, there's so many different things proteins do. Enzymes are proteins. We have protein involved in structure. We have protein involved in so many different functions. Things like antibodies that fight infection. All of those different things are related to protein. And so the importance of getting from DNA to RNA to protein cannot be underestimated. Let's talk about this first part. Transcription. So this term trans means across, script means writing. So I like to think about just writing DNA into RNA. A couple reminders about RNA itself. So we know that RNA is a little bit different than DNA in just a couple ways. For example, we use ribose as a sugar instead of deoxyribose. You're going to use uracil as the nitrogenous base instead of thymine, and it still bonds with adenine. And it's generally going to exist as a single strand. The way I like to remember the difference is RNA loves you because it's uracil, so that's how I remember it. Anyway, the process of of making a single-stranded mRNA by copying part of the nucleotide sequence from DNA, that is called transcription. mRNA. So that little M stands for messenger. There's actually a couple of different types of RNA. We have mRNA, which is our messenger RNA. It's going to carry the message of how to build the proteins. We have rRNA, which is going to form part of the ribosome. We have tRNA, which is going to be important in translation. So we have all these different RNAs playing a role. Okay, so let's go through the steps of transcription really quickly. So first we have all of this DNA inside the nucleus, but translation, the process of turning it into a protein, occurs outside of the nucleus. And so transcription begins when there's going to be this promoter region, some part of the DNA that has a signal that says to copy this. When that happens, there's going to be an RNA polymerase, a protein. It's going to come to that section of the DNA. The DNA is going to be separated, and then after that DNA is separated, half of it is going to be copied via base pairing rules into mRNA. Eventually, the end of it is going to be reached. There's something called a terminator, and it's going to stop being copied. mRNA generally just copies the instructions for one polypeptide. After you have that mRNA, there's going to be some things that get changed about it at the end of transcription. We'll talk a little bit more about those later. But once we have that, we're going to jump into translation. So translation is the process of actually making the polypeptide using that mRNA, also using tRNA in the ribosome. One key idea here is the idea of a codon. So we know there's a group of 3 nucleotides that code for a single amino acid. That group of 3 nucleotides we call the codon. Codons are fantastic because they're universal. The same group of 3 in any organism that we know of is going to make the same amino acid. So there's 20 different amino acid options, and based on how we put those together, we get to those billions of different options of proteins. And this is sort of a little code to go between the idea of nucleotides into amino acids. So oftentimes we'll see this code written in this way. The way you read this is just again in groups of 3. So first you look for what your first base codon is. So let's say you had the codon AUG. So you'd look here and be like, oh, my first one is A. Then you look on the top for your second one is U, so you know you're looking in this box. And then the last one here is G, so you know you're looking at AUG, which is methionine. You're going to see that methionine is going to start every single amino acid. It's called the start codon, and so we'll see a lot of that. Also, sometimes we see a chart like this. This one is read the exact same way, only read 5' to 3' direction. You start in the middle and go towards the outside. So if you had AUG, you start in the middle with A, then you find in that quadrant U, and then all the way out to G, and again we get to methionine, our start codon. Each one of these amino acids has a 3-letter abbreviation and a 1-letter abbreviation. Sometimes you'll see either way. For example, if you have AUG, CUG, you're just going to break this down into groups of 3. So AUG, I read that AUG, there's methionine. CUG, I read that CUG, there's leucine, etc., etc., etc. Eventually we'll get to a stop codon. There's a couple of different options for those, and that's going to be when we stop translating. With translation, there's 3 big steps. First is initiation. With initiation, there's going to be this mRNA, which is represented in this way, and there's going to be a tRNA that has the methionine on it. Notice this tRNA looks kind of like a clover. There's different tRNAs that have what we call an anticodon on them. The anticodon will bind with the codon based on base pairing rules. But the tRNA also brings with it an amino acid, and so based on those base pairing rules, that's how we get the correct amino acid into that spot. This blue thing here is supposed to represent the ribosome. So there's actually 2 halves to the ribosome. Those 2 halves will be separated until they bind during initiation. There's 3 big parts to the ribosome. We call it the E, the P, and the A site, and we'll see that most of this occurs in the P site is when this is going to happen. Okay, after initiation, this thing is going to get longer. So what's going to happen there is the ribosome will move down, and then another tRNA comes in based on the base pairing rules, and then a peptide bond is going to form between the amino acids, and it's going to slide over. So it goes from E to P to A, sliding over as we move down the mRNA to eventually form this amino acid sequence. Finally, there will be a termination sequence. So once you reach a stop codon, then the whole ribosome is going to break apart, the tRNA is going to leave, and you'll be left with a polypeptide. Sometimes those polypeptides can be altered a little bit in things like the ER or in the Golgi apparatus, but that is nearly a finished protein. Okay, so for a really, really long time, there was this idea that if you have one gene, that is just one polypeptide. So one sequence of DNA forms one protein. That's actually changed a lot. So if we look at this graph right here, this is the estimate for the number of genes over time. Notice in the 1940s, we got around 100,000 genes, what people were saying. And then as time goes on, we're going down, 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 down. Notice this really, really big drop here between 2000 and 2005. During that time, we had something called the Human Genome Project. We actually took the genes of a human and we sequenced all of them. And all of the genes that you have, that's called your genome. And in looking at all of those genes, they were actually able to find the actual number of genes is much, much, much smaller. Now the estimate is somewhere around 20,000 to 25,000. Thinking it used to be 100,000 and dropping down that much is really significant. And the reason that's quite possible is because of some different things that we can do with a gene. So say you have a gene and you copy that into mRNA. What you can do is you can cut up that mRNA and take parts of it out. So if here's some DNA and it gets copied into mRNA, and then you slice up some of these and take these blue sections out, the blue sections will be called introns. They're the ones that get removed. And then exons are then spliced together, and that is going to form the actual mRNA that leaves the nucleus and then can be translated into a protein. The idea is that you can have alternative ways to do this. We call this alternative splicing. So let's say you take out just a couple of these ones, these are the introns that are removed, that might create a protein that folds in a different way. Versus if we take out a different intron, it may fold in a different way. A different intron folds in a different way. So we may be able to create multiple proteins from a single gene based on which parts we cut out and which parts we keep. What I think is totally fascinating about that is there's genes that are going to control what things you cut out, and there's genes that are going to control what part you keep, and there's genes that are going to control how all of this stuff works. And so the process of making proteins is controlled by proteins, which is controlled by DNA, and all this stuff is really cyclically related in your body, which I just think is totally fascinating. So hopefully this was a good kind of review, sort of simple beginning towards this stuff, and we'll delve into a little bit more.